Hi there guys, and welcome to another video of A-Level Biology with Mr Phillips. It's another year 12 video from the Biomolecules module. So, what we're going to be looking at today is triglycerides, which are a type of fat, a type of lipid. We're going to look at the structure and how they bond to form a triglyceride. Just to remind you that lipids cannot be classed as polymers because they are made up of two different types of molecule. So what are lipids? Are the group of chemicals that are non-polar, they're hydrophobic, so they will not dissolve in water. They're insoluble in water, but will dissolve in alcohol, which is how their actual food test works. And they're made up of carbon, hydrogen, oxygen, but obviously they have, as you're about to see, they have a lot less oxygen molecules compared to something like glucose, a lot more carbon and hydrogen due to the fatty acid tails. So this is the basic structure, and we'll go into this in a lot more detail, but I thought this was a nice little picture to introduce it. Basic structure of a triglyceride molecule. So you've got a glycerol molecule there, okay, and then the triglyceride bit comes from the fact there are 3 fatty acid chains, and we'll look at how these bonds are formed later on. We have a look at one of the fatty acid chains a bit closer up, what the basic structure of the fatty acid chain is. So sort of at the end of the fatty acid chain, so if I sort of say here, okay, this bit here is what this molecule represents here, okay, and effectively there is a double bond O and an OH, it's a carboxyl group effectively, and at the end of it there is an R group, and that is your chain of fatty acids, and that could be just a couple of carbons in length, it could be huge amounts of carbons in length as we look on. So triglyceride is one type of lipid effectively that we need to know about, and basically it's condensation reactions between fatty acids and glycerols. Just like in previous molecules, condensation reactions bring 2 small molecules together to form a bigger molecule. So these are the 2 sort of structures that are made from. So again, just what we've sort of said before, there is a glycerol molecule, which is a relatively simple molecule, but you do need to know the sort of structure of it. So there's 3 carbon atoms, and at one side of the carbon atoms there is hydrogen, and literally just a hydrogen, and at either end there's just a hydrogen, and then at the other side of all the carbon atoms there are 3 hydroxyl groups, whereas the fatty acids, as we said a moment ago, have this group on the outside and they've got the hydroxyl groups. So for people that watched the previous videos or have already studied a lot of A-Level biology, you will probably have no difficulty in thinking, well, there's going to be a condensation reaction between the hydroxyl group there and between the hydroxyl group there. But this time it's not going to be just one fatty acid. You can have 1 fatty acid, 2 fatty acids, 3 fatty acids, making a triglyceride molecule. So this is the structure of glycerol, as I said. So it's got 3 carbons, it's got OHs attached to each carbon atom, and then on the other side where the condensation reaction happens, then we've got hydrogens at either end and 3 hydrogens along the side. Structure of fatty acids is basically a long chain of hydrocarbons, okay, and that can be different lengths. And then, as we said before, at the end they've got a COOH, or acid group, or carboxylic acid group at the end, and they can be either saturated or unsaturated, which we'll come on to in a moment. So how does a triglyceride molecule react and bond? Well, if you look at this diagram, it shows you pretty clearly how it does it. So as I said a moment ago, each hydroxyl group of the glycerol molecule is going to react with a hydroxyl group on the end of the fatty acid molecule. So effectively, if we look at the bottom one and use that one as our example, it doesn't matter which one you would choose, which oxygen group in an exam, but you've got 1 oxygen and 2 hydrogens, so H2O. So a water molecule is going to come out, and because the water molecule is coming out, it is a condensation reaction, just the same as when amino acids combine, just the same as when carbohydrates combine. And what that leaves us with is the oxygen molecule. So if you think about this example here, we'll be leaving us with this oxygen molecule here. Now oxygen's got a valency of 2, so that molecule's got a spare bond now here. So the oxygen molecule goes over, if we look over here, the oxygen molecule has got this bond and this bond now, so it's adjoining the glycerol molecule to this fatty acid, and the same happens above, this one to this one, and again to the top one as well. Now that is obviously using the removal of water, so again condensation reaction, and it forms a covalent bond. But again, if you remember in the exam, don't call it a covalent bond, you need to call it by its specific name. So sugars, it's a glycosidic bond, amino acids is a peptide bond, and in fats and lipids it is an ester bond. So it's an ester bond that is used to make it. And obviously we wanted to break that molecule down because it's a reversible reaction. If we wanted to break that molecule down and split it back up, we would just add the water back into this bond, and that would then break it down in a hydrolysis reaction. So just a bit more about fatty acids then. So effectively we've got our fatty acids at one end of the fatty acid chain, opposite way around to how I've been showing you earlier before. There is the end of the chain which would be a CH3 group, which is called a methyl group, and at the end of the chain where we've just done the reaction, there is the carboxyl group, okay, which is obviously originally before they bind has got the OH there, or the carboxylic acid group, or the acid group. And then we've got the long chain of hydrocarbons in the middle. Now, in terms of what else we need to know about that, that is pretty much it. But there is the chain, the tail, the hydrocarbon tail of a fatty acid that is hydrophobic, so that bit repels water, whereas the head of the fatty acid, if you think of it like that, the carboxyl group, has a hydrophilic level which is attracted to water, which gives fat its properties of being insoluble in water. So there are two types of fatty acids which we need to know about. Animal fats, which are generally solid at room temperature, are what we call saturated fatty acids. Now, what saturated fatty acids means is that every carbon atom in the chain is just single bonded carbon bonds, okay, and that gives the specific properties that makes it have a higher melting point, which makes it a solid generally at room temperature. Unsaturated atoms, which make up plant oils, so something like olive oil or sunflower oil, they've got a double bond carbon, which means that they are unsaturated and have a lower melting point, and that means they're liquid more often at room temperature. Then there could be something called polyunsaturated, which is obviously very healthy for us, and that is where you've got lots of double bonds. So what is the difference? Well, this is the basic difference. And effectively, if you've got a saturated fatty acid, so as I said, animals, for example, you've got just single carbon bonds, okay, no double bonds, and that gives you a much more straight fatty acid tail. Every time there's a double carbon bond in the unsaturated fatty acids of plant oils, you get a kink in the tail. And what this means is that you don't get straight chains anymore, you get kinks in the tail, which could alter the shape of the fatty acid. So why is that important? Well, that's important because if you get straight chains, so go back to animal lipids again, so animal fats, okay, so animal fats are completely saturated, there are no double bonds, and what this allows is that the fatty acid chains can get really close to each other. So one fatty acid can line up next to the next fatty acid, which can line up next to the next fatty acid. Now what that does is it means if they're getting closer, if they can stay close to each other, it's going to mean that they're going to be held together a lot better, and therefore they have a higher melting point, which means they're generally a solid at room temperature. Whereas whereas if they are unsaturated fatty acids such as plant oils, it's quite hard because of the kink which is caused by the double carbon bond, which alters the orientation, alters the angle of one carbon atom bond to the other one. It's quite hard to actually have them close to each other, because they'll have a double bond. Let's say there's one here, okay, and that could alter the structure, so then it goes down here. So they can't lie close to each other, and as a result they have a low melting point and tend to be more fluid in nature. And that is really important as well, just mentioned with a membrane structure. So if you want a more fluid membrane, you will have some unsaturated fatty acids in the membrane, as opposed to a less fluid membrane, which will have just saturated ones. But we'll come on to that when we do about membranes in a few videos time. And the last bit is the the test for lipids, the biological molecules test, or the food test as you would have called it at GCSE. So basically this is called the emulsion test. And what we do with lipids is, because lipids are soluble in alcohol and ethanol and insoluble in water, we can do the emulsion test. So what we do is we take our sample of lipids and we dissolve them in ethanol. Now what that means is the lipid will dissolve, it will go clear, and because the lipids are then insoluble in water, when we add water to that mixture, the lipid which had dissolved will come out of the solution and will become a precipitate. It will sort of solid within the solution, and what you'll get is a lipid layer forming of a white cloudy emulsion forming where the lipid is. If you've got a negative result, if there are no lipids to be formed because it isn't lipids in your sample, then you get no white precipitate forming. And here's a picture of it. Normally, because lipids, don't forget, are less dense than water, they will also float at the top of the solution as well. So sort of two methods you can see in one test. It's got to be a lipid because it's less dense than water, so it's floating on the water, and it is insoluble in water. So when you put in the alcohol, originally it dissolves, and then you put the water in, and that causes it to come out of the solution as a precipitate. So here's some questions for you. We'll do next video where we'll look at phospholipids, just to summarise the lipid section up. Thank you very much, and I'll see you soon.